thanks everybody for coming. For those of you who don't know me, I think I know most of you, but I'm Lance Bollinger. I'm one of the faculty here in KHP. Grace is actually my first master's student doing a thesis, so we kind of want to share with you kind of what she's doing the past year or so. And this is actually something she's going to be presenting at Southeast ACSN as well. She was accepted to be competing for one of the student awards there. So we're trying to get a little practice run, questions, answers as far as what she's studying. But essentially what we're looking at is the effects of vitamin D on skeletal muscle lipid function. Anything else there?
Okay, good morning. Thank you for coming to my thesis presentation. So this is what I've been working on since last fall. It's been a long time coming, but I've learned a lot, so that's a plus. So the title is Calcitriol favorably alters lipid partitioning in skeletal muscle cells. So we're going to be looking at how calcitriol affects lipid function and lipid partitioning in the muscle. What is the role of vitamin D in skeletal muscle and how is that affected by exercise? So calcitriol is the active vitamin D metabolite in your body. So that's what I'm referring to when I say calcitriol. So for starters, what is vitamin D? How does it work in the body and muscle? So vitamin D promotes proper calcium absorption to aid in muscle contraction and just overall calcium homeostasis in your body. It's very important. It increases mobilization of calcium from the bone into circulation. Again, very important. And it also increases reabsorption of calcium by the kidneys. Just kind of three big factors, or I guess you can say the most important factors of vitamin D. A little more background. So I think we can all agree that proper storage and utilization of lipids within the muscle is essential for quality of life, energy, overall health in general, using them most efficiently, I should say. Circulating 25-hydroxyvitamin D3 is also been shown to be directly correlated with IMCL content, intracellular lipid content. 25-hydroxyvitamin D3, 1-alpha hydroxylase. That is what actually hydroxylates 25-hydroxyvitamin D3 to give us calcitriol in our bodies. So that step is really essential to actually get calcitriol's main active form, or the active form of calcitriol. Calcitriol mediates its effects by binding to its vitamin D receptor, or VDR. VDR is probably one of the biggest key players. It's essential. You have to have it to have absorption and other downstream effects. It is a nuclear receptor to obviously induce or repress gene transcription. A majority of the lipid research on that side of my thesis has been looking more toward tags and DAGs, just other lipid forms, and ceramides, again, with perilipin proteins, and the perilipin proteins are what actually coats the lipid droplet. So when I refer to pelins, tags, DAGs, that's what I'm talking about. So this picture, I kind of like it in the form to show how lipids work in skeletal muscle, what's their purpose, why do you need them. So we didn't specifically study adipoconnectin, so I don't want you to get too transfixed on that. But when you think of adipose tissue, you can think of the fact that it stores energy in the form of lipids, and that's essential for our function in the body, especially with vitamin D. But it has all these downstream effects, which are great. They're all essential with and without exercise. Glucose translocation with increased glucose uptake. Obviously, you need that as well. Increased fatty acid oxidation, which we look at later, or we discuss later, I should say. And increased fatty acid uptake, all of which can be used for energy and other daily functions in the body. So it definitely plays a role, lipids definitely play a role in skeletal muscle as well. That's how they're connected. So, Dr. Thomas, Maya, this graph might look familiar to you. This is previous research done on the correlation between IMCL, internalized cellular lipid content, and vitamin D in the gas drop. And as you can see from this graph, they saw a positive correlation as vitamin D increased, so did IMCL in the gas drop. So they are related. Something going on there. So what is vitamin D? Why we're looking at it, what is its purpose? It is a regulator of bone metabolism and it's needed for absorption of calcium. Vitamin D3, the prohormone, is produced in the skin after exposure to sunlight. So we know we get vitamin D from the sun, that's why it's important. Vitamin D3 is hydroxylated in the liver to 25-hydroxy vitamin D3. And the 25-OHD3 is then hydroxylated to the active hormone, 125-dihydroxy vitamin D3, which is cholesterol, as we say. So that acts on its very important receptor, the vitamin D receptor, or VDR, which leads to potential influence on other pathways for protein synthesis, protein degradation, those other downstream effects that we were talking about. So this picture, it kind of just describes 125D cholesterol effects, both indirectly towards the left and directly towards the right. We have this VDR, the receptor like I talked about, and it's RXR complex. And of course it has genomic effects to increase or decrease gene transcription, which is where you get your latter downstream effects from. And that, like we said, can lead to contractile proteins in the muscle, which can help contraction, muscle differentiation, and phospholipid composition. And that can lead to improved muscle form, function, metabolism, better storage. There's a bunch of effects written on here. The right side probably has more to do with today's presentation. But obviously, systemic calcium and phosphate homeostasis is extremely important. And that as well leads down to improved muscle function, improved contractile activity, all the essentials that you need, or that we need. So I added on to this image that kind of just displays whole body level of why it's important or why it's needed. Lipid partitioning and lipid packaging, which is kind of the other side of my thesis. So what do we mean when we say lipid partitioning, lipid packaging? That's when we talk about our lipids that we studied. So DAGs, tags, and ceramides, they're all species of intramyocellular lipids. The main players that we looked at with data collection so far. So lipids are commonly stored within skeletal muscle as intramyocellular tag, or IMTG, within the lipid droplet. DAGs are lipid intermediates that accumulate in the cell during incomplete tag hydrolysis. So it's just a different form. You can see all the variations here. Here's a diglyceride, here's a triglyceride, tri. See the different bonds there. And then lastly, ceramides are a type of bioactive pseudolipid found within the cell membrane. They can act as a secondary messenger. So yes, they're all lipid subspecies, or not subspecies, intramyocellular lipids. They all kind of have different and changing roles, and we saw that with some previous data questions. So we'll talk about that in a little bit. These images over here are just so you can kind of see the variation in chemical structure and how they shift from the glycerol backbone. Does that make sense so far? Okay. So the perilipins, or the P-lines, as I mentioned at the beginning, these are what coat the lipid droplet, and you can kind of think as these guys as the packaging proteins. So they associate with and coat the droplet. like I said, and certain ones either promote lipid storage or lipid lipolysis. You can think of lipid oxidation of the lipids that they're putting. So there is PLN 2 through 5, and they're all expressed in skeletal muscle, which was important for us to know. PLN 2 and 3 promote lipid storage, and you can obviously probably think that expression of those is increased when the cells go through lipid overload or are fed high amounts of lipid. And we've seen that, or I've read that genetic knockdown of PLN2 in C2C12 cells increases cellular oxidative capacity. So without that storage protein there, you could have increased cellular oxidative capacity, which makes sense because that one's originally for storage. And then PLN5, otherwise known as the oxidative PAT protein, is expressed in highly oxidative tissues during states of increased fatty acid oxidation in skeletal muscle. So when you think PLN 2 and 3, you can think lipid storage. When you think 5, you can think lipid oxidation. Does that make sense? So other important lipid metabolism enzymes to kind of inform you guys about. Hormone-sensitive lipase, or HSL, is the key enzyme in the regulation of lipid stores. So it's essential there for the degradation of tags to DAGs and free fatty acids. So just another part of lipid oxidation. That goes along with ATGL, adipose triglyceride lipase, which catalyzes the rate-limiting hydraulic step of tags in that lipolysis sequence. So again, needed in the lipolytic side. And then you have DGAT, which is actually the other portion of creating more tags, or catalyzing the formation of more tags from DAGs and acetyl-CoA. So this image over here kind of helps describe that, tags to DAGs and then to monoglycerides. You can kind of just see, it points out how each enzyme works in the sequence of the shift from tags to DAGs or DAGs and tags. Like we said, DGAT, formation of more tags. So DGAT, DAGs, tags, more tags. So that's an example. And that's all I'll say about that. So, kind of bringing it back around, what's its relationship between, what's the relationship between exercise and lipids? What has already been researched? What is the point? Acute exercise actually increases lipolysis from tags, and chronic exercise training increases storage of IMTGs. That would kind of support the fact that it might be associated with improved oxidative capacity or improved muscle function that comes behind the improved oxidative capacity. Following endurance and sprint interval training, protein content of PLN3, so storage PLN, and OXPAT, which is the oxidative one. You can kind of think of it as a oxidative OXPAT. Increase in human vastus lateralis muscle. And that kind of helps us support the fact that these proteins do respond to exercise training, which was kind of an important question that we were thinking about in thesis development originally. So lastly, reported correlations have been found between skeletal muscle oxidative capacity and OXPAT content following endurance exercise. And this kind of suggests that OXPAT is involved in the regulation of fatty acid oxidation, which is important when you are trying to oxidize those fats or you're going to use them for energy. So now coming into my specific aims and hypotheses so far. My specific aim is to determine the effects of calcitriol on mitocellular size and intramitocellular lipid accumulation and mobilization. My first hypothesis is that exercise increases expression of the genes involved in vitamin D metabolism. one is that calcitriol increases myocellular size and protein synthesis. And my last one is that calcitriol modifies lipid partitioning, packaging, and mobilization. So, now kind of diving into what we're working on, or have been working on after all that background. Hypothesis 1, so like I said, exercise increases expression of those genes involved in whole vitamin D metabolism. So we are working with C2, C12 myotubes, so cells from mice. Didn't have to go through IRB for humans, which is good. The cells undergo one 30-minute bout a day of cyclic stretch, and I'll show you that in a second. So it's kind of like a chronic exercise idea that we're working with. And results analyzed by tests. So this video, if it'll play, look at those movie stars in the back. Hey, Dr. Lauren. So this is our flex cell system. Can you guys see? Ooh, so exciting. So I'll play it again just so you can kind of see a better visual, but the bottom of our wells that we use for our plates, so there's 6 wells. The bottom of them is kind of like this flexible plastic, so if you like push up on it with your finger, you can move it. And these little white pieces, with the help of a vacuum pump that comes into this incubator, it sucks air in and out, and these little white pieces push up on the bottom of our wells, and that stretches the bottom of the well, which then stretches the cells that are on the bottom of the well. That's what we mean by cyclic stretch. So I hope you can kind of see it this time, maybe a little better. You guys see it like pushing up? I hope you can see it. Everyone can see it. Super awesome. So that is what we mean by cyclic stretch. It's a flex cell system, hence flexible bottom well for the cells. So some data that we've kind of seen already from, or that we've collected already, are how cyclic stretch is affecting the myotube protein content and protein synthesis and degradation. So when you think of this stretch system, you hear me talk about it more today, you can kind of think of the idea, think about mechanotransduction instead of us really exercising the cell. It's more of a the cell being stretched versus exercise or contracted. It's more of a stretch. So this idea of mechanotransduction should kind of stick in your head when we think about the stretch system. So if you look at B, C, and D first, you can see, or B and C, excuse me, myotube protein content and protein synthesis rate increased. So you can see the control versus stretch, it increased significantly. And with the protein synthesis rate, on the bottom here you have the time post 30-minute stretch. So 1.5 hours post, 3 to 4.5 hours post. You can see that the synthesis rate continues to increase. So obviously the cyclic stretch is having an effect there just by itself for those 30-minute bouts for 5 days. And then if you look in the top left, when we're talking about CYP27B1, that is a gene that encodes kind of the formation of calcitriol. So it kind of leads us to think that maybe it has kind of a mediating effect on protein synthesis with the stretch condition at least. So it's just another important factor in calcitriol. So methods for hypothesis 2. Calcitriol increases myocellular size and protein synthesis. So we have C2, C12 myotubes treated with 100 nanomolar of calcitriol. That is what the dosage that we decided to give. Just for your information, it's a pretty high dose, more so than what we see in the literature. literature, but that's the dose that we went with. And for looking at protein synthesis, specifically with our cells, we looked at a radiolabel amino acid incorporation. So we fed the cells tyrosine for 2 hours and then collected the proteins and looked at the radioactivity amount there in relative to the protein there. So that's how we look at protein synthesis. And then protein content was looked at with a VCA assay, which looks at the total concentration of protein in our wells, or in our solution. And those results were in our SPI test. So this first graph, we're looking at protein content and synthesis rate. No significant changes here for 100 nanomolar of calcium treatment, as you can see on the bottom. Just to inform you about no change in protein content, protein synthesis rate there. But so moving on to methods from hypothesis 3. Calcium trial modifies lipid partitioning and packaging immobilization. So our C12 myotubes here, again, were treated with calcium for 4 days, 100 nanomolar. And they were then delivered to Dr. Andrew Morris. He's over in the BBSRB building. Him and his BioCore mass spectroscopy lab analyzed our cells with LCMS, which is liquid chromatography mass spectroscopy. It's a lot to say, but it kind of just breaks down what is inside our wells and separates, helps us to analyze, quantify our lipids and kind of see what is there. So it's a really important step for us to see our lipid data. But then real-time PCR to look at the mRNA content in our cells was performed 24 hours post-calcium treatment, so post 100 nanomolar. So with this is where a majority of our lipid data you'll see has been analyzed, and our mean tag content was not significant. And on the other hand, if you look at our DAG content, diacylglycerols here, the total was not significant, letter A. But if you look at B, C, and D, these lipid subspecies, they all, minus 16.0, or 16.0, excuse me, increase significantly, showing that increase in DAGs. So when you guys see, this is like traditional lipid nomenclature, the way it's written. The first number is the number of carbon atoms, and the second number is the number of double bonds, or the number of unsaturations, saturations. There for the subspecies itself. So that's what we're talking about when it looks, it's written in COP, but obviously it's not. So no change in total there, but you see changes in the intramicellular lipid subspecies. So you might not have a shift in total, but something else is obviously affecting the intricate subspecies. DAG content for 18.0, or 18.0, excuse me, is, so 18.0 also, I forgot to say this, that is oleate, whereas, if we go back one, 16.0 is palmitate. So palmitate is a really saturated fatty acid. I'm not sure if you have heard of it. Oleate is unsaturated. Unsaturated tend to obviously be better for you instead of a saturated fatty acid. But this mixture right here, DG16O18.1, is palmitate and oleate, the mix. So just want to let you know that. So we'll talk about it later. this is how lipids are generally seen, at least in the subspecies format. But 18.0, so that oleate-based fatty acid for JAGs increased with calcitriol treatment. Same thing with 18.1 and 20.0. So moving on to ceramides, just like total DAG content, total ceramide content was not significant. Ceramide 14, 16, and 20, so just other subspecies of ceramides themselves, all decreased significantly, which is a little different from DAGs, but they're also different lipids. So coming into this idea of lipid packaging, these proteins, or P1, 2, 3, and 5 oxbat, and BDR, the gene. This is a graph looking at the gene expression of all these guys, which are important players when we're looking at lipids themselves and vitamin D function together. So BDR, our vitamin D receptor, went up with calcitriol treatment, and P1-2 and 3 did as well significantly, and then we did not see a significant increase in the oxbat or the oxidative one. So this kind of leads us to think or believe that you have this increased lipid storage due to the fact that 2 and 3 significantly increased, and we didn't see a change with oxbat. So that was beneficial to us to see kind of what the variation was there between the storage ones and the oxidative ones. Storage proteins, oxidative proteins. Next, when you think about, so remember talking about HSL and HCGL for more of the lipolysis side of it all, and then, or you can think lipid mobilization, and then DGAT for more of formation of more tags, so more lipid storage. DGAT significantly increased, and that was the only one when we were looking at the gene expression with calcitriol treatment. DGAT was the only one that significantly increased, again, to make you think that there's increased lipid storage there over increased lipolysis. So a summary of the results that all of these graphs that I've just shown you, kind of bring it all together. We did not see a significant change in mean tag content. What we did see is a significant increase in DGAT, which is, like we said, for the formation of more tags. So it's kind of interesting. We're still studying it, but it was interesting to see a change, a significant increase in DGAT. The isoglycerol responses, or the DAGs, were a little more varied. So calcitriol significantly increased this palmitate oleate structured mixed fatty acid. Remember me talking about that with calcitriol. And then calcitriol plus stretch significantly decreased the palmitate structure under the 16.0 DAG. And then calcitriol increased DAG content with calcitriol. I just list all the ones that increased from our data. And ceramide content decreased in the individual subspecies, not in total. There was no significance in the total ceramide content, but those individual subspecies did. And there's an increase in the lipid storage perilipins, so an increase in those proteins that affect lipid storage. So how does this kind of relate to what other people are seeing in the literature and what other people have found? So Brett Goodpasser does a lot of great work looking at lipids and insulin resistance. And I think some of you, if not all of you, have heard about the athlete's paradox, especially when talking about endurance training athletes. But this paper just kind of is a nice comparison to the data that we have found. And if you keep your eyes, so I put a red box around the NWA group, which is the endurance trained group in this study. There was an obese group and a sedentary group, and then a highly endurance trained group, which is, again, NWA. So if you look at their total DAGs, the top left in A, you'll see that the endurance train group had the highest amount of DAGs compared to their counterparts. And if you look at the breakdown down here, so again, NWA, the highly endurance train group, with the 16-0-18-1, so the palmitate oleate structure mix, and then the 18-0, which is oleate by itself. You also see increases in these fatty acids in the highly endurance trained group. And highly endurance trained athletes in general have a high amount of lipid in their system anyway. So that kind of supports that fact, which again supports the athlete's paradox. And then this is another one of the graphs talking about ceramides. So see all the C's here. Again, if you find the NWA, the white box, you can see that ceramide content decreases or generally trends to decrease in this graph, which is also what we saw with our study or our experiment. So kind of relevant conclusions from that study, how it relates to ours. Endurance athletes, the NWA group had an increase in total and mixed DAGs. And again, when I say mixed DAGs, that means one fatty acid saturated and one unsaturated. Palmitate oleate, like I talked about. And that supports our claim as a favorable adaptation. Endurance athletes also in that study had high levels of the palmitate oleate structure, and we saw that same thing with our calcitriol treatment. So that was a strong comparison. And then with their ceramides in this study, the NWA athletes had low levels of total unsaturated and saturated. And that kind of leads us to think that any reduction that you saw with the ceramides there, or increases in the mixed DAGs, seems to be a favorable adaptation, and that's what we want to see. So kind of bringing it back around, this is a little figure I made of kind of how we think it works. And if you look at the top, you'll see 125 OH2 D3, so calcitriol coming in. So there's a cell membrane line. So you go to the cell membrane cytosol nucleus. So you'll have increased calcitriol coming into the system. You will then have increased intracellular calcium. It'll find that balance there. You'll have increased VDR, because that's a receptor used to work through the body and to mediate its effects. You could have increased NFAT signaling, which is a pathway that works by increased calcium signaling. So that could be like a side effect. And then you have translocation of BDR into the nucleus to mediate its effects on gene transcription, which is essentially how you see the rest of the downstream effects. And then you have gene transcription, hopefully, in the nucleus to kind of mediate the rest of its effects in the body. So the lipid droplets, right here, obviously. But what we think is hopefully kind of happening is that you have this increased or repressed gene transcription, and that is affecting the lipid droplet in the way of the paralipin proteins and the DGAT enzyme, all dealing with lipid storage. Does that make sense? So you can have hopefully this increase of all these to promote lipid storage, but then to also kind of break it down into these kind of smaller lipid droplets, like more of them, with that increased storage. Does that make sense? So overall conclusions, calcitriol does not affect mitocellular protein synthesis, but favorably alters IMCL partitioning within skeletal muscle cells. And it increases expression of genes involved in IMCL packaging and storage. So PLN2 could be DGAT, just like we talked about in the last slide, in that lipid droplet, coating that lipid droplet, affecting it. And it seems to have greater effect compared to cyclic stretch this last picture is kind of just a simple breakdown of what I've been saying the whole time. But the top part is just how vitamin D is hydroxylated and how it comes to have its effects on the kidney, bone, macrophages, and then on the muscle, which is what we're looking at, what we're studying. So its effects on muscle, gene effects and self-signaling, which is what you hope for. And then I drew an arrow out here to say like an increase in lipid storage in TGAT. That's what we've seen and that's what we think is happening. But then the image itself kind of draws arrows down to muscle hypertrophy and strength, which has also been studied, and then improved muscle performance and reduced falls, which is what a lot of people think of when they think of vitamin D, especially in the elderly population. But I also have this box, altering oxidative capacity, which has a question mark after it because we don't fully know the effects there. So it's kind of a thought that we're looking at, how could it or does it alter oxidative capacity there? So have these great targets over here that vitamin D works on in the body to maintain calcium homeostasis and just beneficial overall health. And then you have the muscle side where you would hope to see these gene effects and cell signaling, which we have specifically seen on the storage side of lipids. So practical application, what kind of the side of who cares, how can you use it? Increased vitamin D may alter lipid partitioning to enhance muscle performance. Like we talked about with the Good Pastor study, all of the effects they saw, they claimed as favorable effects. I think essentially they can be. If you think about increased oxidative capacity with the change in storage and increased reliance on fatty acid oxidation. So again, question marks after it because it's kind of how we think it could be applied, not necessarily a proven thing, but secondly, the effects in a human model, like taking our study to a human model, are still unknown. And cancer cachexia is another thought, and Dr. Thomas is actually about to look into that with and without exercise in older adults, correct?
Cancer patients.
Cancer patients. They're still older cancer patients?
Not necessarily 45 enough.
No, okay, my bad. But yeah, so he's about to start studying that too, and not like you haven't been studying vitamin D for a long time now. But so that could be another side effect as to how it could be practically applied outside of just C2, C12 matches. So future significance and directions that we hope to take besides the cancer cachexia route is to provide a deeper understanding of lipid packaging and utilization in skeletal muscle, which is really important. Also to aid in understanding the roles of vitamin D metabolism and skeletal muscle function, which could potentially influence pharmacological intervention, the way people supplement or see or use vitamin D, just to kind of understand the better roles of it. And then human and dietary experimentation with varying dosages of vitamin D supplementation to see maybe how a lower or higher dose affects muscle function and muscle performance with aerobic models and anaerobic models, so both sides of the exercise chain there. So my last slide, probably one of my favorites. My biggest credits, Dr. Bollinger, my advisor. You get a huge round of applause. And then the rest of my committee, just extremely thankful. Dr. Abel, Dr. Thomas, Dr. Peterson, who could not be here. She's in the College of Health Sciences. Dr. Andrew Morris, who did our LCMS data, extremely thankful for him, and then Adrienne Herrenbruck, which she's a doctoral student, but she's helped us out tremendously in lab. So huge thanks to all those great people, and that is all. other comments?
Can you go back to your practical application slide? So, I think my question here is that, you put that vitamin D may enhance muscle performance and increase oxidative capacity. You really didn't show those data, right?
Yeah, it's not, and that's why we have a question mark after it. It's kind of like a thought that we have going into the finalizing of data collection and just thinking like, where could it go?